Hi everyone, welcome to chapter 4. This is going to be a really condensed chapter. We're not going to be covering most of what is found in chapter 4, and the parts that we are covering, we're not going quite as deep as the book is, so you can kind of take this chapter as a bit of a breather, although the material that we do cover is going to be very important. So let's start with our not really attendance questions for the day. If one side of DNA strand is AACGTA, what is the other side? Next, what organelle makes protein? And third, where is DNA found? So go ahead and try to answer these questions. They're going to be relevant to what we cover today. And hopefully you've paused your video and tried to answer these questions, but let's see what the actual answers are. If one side of a DNA strand is AACGTA, what is the other side? Well, we're going to use our base pairing that we learned about at the end of chapter 2. And remember, A always pairs with T, adenine always pairs with thymine. C always pairs with G. Cytosine always pairs with guanine. So the opposite side of this DNA would be TTGCAT. What organelle makes protein? That would be the ribosome, and this could either be free ribosomes that are loose in the cytoplasm, or it could be ribosomes that are bound to the rough endoplasmic reticulum, depending on which protein we make and what the goal of that protein is. And where is DNA found? DNA is found inside every cell in our body, and it is found inside the nucleus. The nucleus, remember, its function is to house and protect the DNA. DNA, we're going to see today, is extremely important. We have to do everything we can to protect the DNA. So we keep it packed tightly away inside the nucleus, but then there's going to be even extra measures on top of that to make sure that the DNA is safe. So first, let's kind of refer you back to chapter 2 at the very end when we were looking at organic macromolecules, DNA and RNA structure and function. We're not going to go through and refresh that. I am just going to refer you back to chapter 2 if you're a little foggy on this. Hopefully it's still pretty fresh in your mind because it wasn't that long ago. But if you're not really comfortable with this, go back and listen to the last lecture of chapter 2 when we discussed the structure and function of DNA and RNA. But looking at DNA, now we're going to start looking at a little bit more of the structure and the function of DNA in more detail. Now, DNA, like I said in our not really attendance questions, it's extremely important, and your cell goes to great lengths to protect that DNA. Because if anything was to happen to that DNA, then that will interfere with protein production, it will interfere with you at a genetic level, and can lead to all kinds of problems. So, we've already seen that DNA is stored inside the nucleus, and that the nucleus, that double layer envelope that lets very few things in or out, that is one method to keep the DNA safe. But DNA is actually not just this loose strand of nucleic acid. While it is inside the nucleus, most of the time it's also going to be compacted into something called chromatin. Chromatin is DNA that is also further protected by being wrapped around histone proteins. And histone proteins is what we see right here in this image. Histone proteins are a few proteins linked together, and then DNA is wrapped around it and chemically bound to those histone proteins. So why would we want to do that? Well, DNA when it is loose, there is exposed portions that potentially other chemicals could attach to. And that's not something that we want. So what we could do is wrap the DNA around these histone proteins, chemically bind the DNA to those histone proteins. And now less of that DNA is exposed. So there's less likelihood of something binding to it. But let's go even further. Let's take that DNA wrapped around the histone proteins and kind of, if you've ever taken a string or something and held one end in one hand and one end in the other and twisted the ends of it in opposite directions, it starts to get to a point where it's so twisted it starts to kink on itself. Well, DNA does that. Here's the DNA wrapped around the histone proteins, and then it's going to start coiling back and forth on itself. And the purpose of that is by coiling back and forth on itself, it becomes really compacted, and even less of that DNA is going to be exposed. So there's even less likelihood of something binding with it. So most of the time, DNA in your cells is not just in the nucleus, it's also condensed into chromatin as a protective mechanism to keep things from binding with it. Now, in our cells, we have a complete set of DNA in every cell that we have. Now, that DNA contains all of the genes of our body. And we'll talk about exactly what genes are here in just a few minutes. But every cell has a complete set of DNA. It's just different cells have different parts of that DNA turned on or turned off as needed. So the DNA is not just one really long strand. It's actually going to be multiple long strands, and each of those strands is called a chromosome. A chromosome is an individual specific strand of DNA. And in humans, we have 23 of these, 23 chromosomes. Now, we number them chromosome 1, chromosome 2, chromosome 3, all the way through 23. And each of those chromosomes will contain a specific set of genes. Every human on chromosome 1 will have the same types of genes. They will not have the same gene, but they will have the same types of genes. So the gene for a specific protein will be found on the same chromosome for every human. Now, normally, throughout a cell's life, the cell is not going to need to make a copy of itself, which we will see at the end of this lecture. But if a cell needs to make a copy of itself, if we need to go through cell division, well, one of the things we have to do is completely copy all of the DNA in that cell. Now, when we go to make a copy of the DNA condenses even further. So it goes from chromatin and is going to be compacted even further. And when it is at its most compacted, we see something like we see down here at the bottom. And in lab, when you looked at your onion root tip and you could see these little things that looked like worms, the chromosomes of that cell, that's what you were seeing, that really, really highly condensed DNA. But since we've made a copy of it, there's not just 23 chromosomes. There are going to be 23 chromosomes plus another 23 chromosomes because we completely duplicated all of them. And when we do that, that means we're going to have 2 copies of chromosome 1, 2 copies of chromosome 2, all the way through chromosome 23. Now, when we do that, the copies of chromosome 1 are going to find each other and pair up, like we see down here at the bottom right, or like we see here in this middle image. On one side, we have a copy of, for example, chromosome 1, and an exact copy of chromosome 1, and they're going to be held together. These two copies of the same chromosome are called sister chromatids. Sister chromatids are duplicates of a single chromosome. So if a cell is going to divide, there's not going to be 23 chromosomes. There are going to be 23 pairs of chromosomes. And the pairs are going to be held together at a region called the centromere. The centromere is this pinched in area where the two sister chromatids are joined together. And on each side of the centromere, there's going to be a little structure called the kinetochore. The kinetochore does a couple of things. One, it helps as part of the centromere to hold the two sister chromatids together. And second, when the cell goes to divide, the kinetochore is going to help the DNA to be moved from place to place around the cell. So later when we're talking about mitosis in this lecture, remember the kinetochore is going to help with this DNA being moved around the cell. Now let's go ahead and look at some of the functions of DNA. We're going to talk about genes. Now we've all heard about genes, but most of us probably don't know the definition of a gene. And we're going to look at two definitions that we can use in this class for genes. Well, the most straightforward book definition of a gene is a region of DNA that codes for RNA. A region of DNA that codes for RNA. But let's be a little more vague. Let's give something that makes a little bit more sense, maybe. DNA contains genes, and genes are a section of DNA that tells your cells which protein to make and when to make it. Genes tell your cells which protein to make and when to make it. Sometimes I like to say that genes are the recipe for protein. So let's see how genes make protein. Well, it's going to follow something called the central dogma of biology, and that's what we see over here on the right. The central dogma of biology. Well, a dogma is just a summation or a way to kind of give the broad idea of a subject. So the central dogma of biology just means the idea that all biology follows. And that's what we see here. DNA to RNA to protein. Well, what does that mean? We take DNA, we make a copy of it as RNA, and the RNA is read to make protein. Every living thing on the entire planet follows this exact scheme. We start with DNA, we make a copy of it as RNA, and from that we get protein. There are no living things that follow something else than this. So what we're going to spend the next few minutes talking about is how does this work? How do we go from DNA to RNA to protein? Well, let's learn some terms. Let's learn about something called a triplet. A triplet is 3 letters or 3 nucleotides. Remember our A, C, T, G that was in, just long strands along the DNA? A triplet is 3 of those letters, 3 of those nucleotides that codes for a specific mRNA. mRNA, messenger RNA. Now, if that doesn't make sense, don't worry, because we're going to apply that in just a couple of slides. But for right now, the definition, a triplet is 3 letters of DNA, or 3 nucleotides of DNA, that code for a specific strand of mRNA. And when we're talking about genes and DNA and RNA of various types, we're going to fall back to something we learned at the end of chapter 2, and that is base pairing. Remember, when we're talking base pairing, A and T always go together in DNA. C and G always go together in DNA. And also we said that in RNA of any sort, there is no T, there is no thymine. Instead, there is uracil, U. So anywhere where we're looking at a strand of DNA, if we have an A of DNA and we want to pair it with a piece of RNA, well, we could put a T there because A and T always go together, but that would be wrong. Remember, RNA doesn't have thymine. So instead, we would have a U. A and U go together when we're talking about messenger RNA or any of the other types of RNA. Again, if this is not making sense, don't worry, because we're going to be applying this in just a few slides. So now let's talk about RNA, and there are lots of types of RNA. We're only going to talk about a few of them. Well, RNA, remember, has some of the same features as DNA. It's still a nucleic acid. It's still made of nucleotides. But it's single-stranded rather than double-stranded the way DNA was. And when we're talking about RNA, we have mRNA, messenger RNA, tRNA, transfer RNA, and rRNA, ribosomal RNA. So let's see what the different types are. Messenger RNA is the RNA that carries the instructions of DNA. It carries the instructions of DNA. mRNA is what we make off of the DNA when we're going to make a protein. And the way that we do that is we take the double-stranded DNA and we unzip it. We just look at one side of that DNA. Remember, DNA was double-stranded. All we do is just take one of those strands away. And we have some letters of DNA. Let's say one of these is an A, C, C, G, T, A, C. And I just made those up. They're not any significance. We're just doing this to illustrate. Well, if this is DNA, and we're talking about messenger RNA, well, first we unzip the DNA, and now we're going to build our mRNA off of this DNA strand. And we're going to kind of follow that base pairing that we talked about. We're going to add one letter at a time. Well, if we have an A on one side, what do we have on the other? Since this is RNA, well, remember, we can't put a T there, even though A and T always go together when we're talking DNA. Instead, we put a U. And if we have a C on the DNA side, what do we lay down? G. And again, C on the DNA side, G. If we have a G on the DNA side, what do we lay down in the RNA? C. If we have a T on the DNA side, what do we have on the RNA side? A. There's an A on the DNA side again, so it's a U on the RNA side. And C gives us a G. So we take the DNA and we build the messenger RNA from it. Well, that's what the messenger RNA is and where it comes from. And remember when we were talking DNA, we said 3 letters of DNA was a triplet? Well, the corresponding 3 letters of mRNA is called a codon. A codon, we're going to give 2 definitions. A codon is the corresponding messenger RNA for a triplet in DNA. But the better definition of a codon is 3 letters of messenger RNA that code for a specific amino acid. There's a word we haven't heard for a while. A codon is 3 letters of messenger RNA that code for a specific amino acid. Now, if you don't remember what an amino acid is, go back and review proteins from Chapter 2, the very last lecture of Chapter 2. Remember, amino acid are the monomers for protein. Amino acids, when they're strung together, they fold up to make a protein. Now we're starting to put together the whole DNA, RNA protein. If we have 3 letters of DNA, a triplet, from that we can build 3 letters of messenger RNA, a codon. What does the codon do? It codes for an amino acid, and multiple amino acids will eventually give us a protein. So we're getting there, and we're going to start to put this all together here in just a moment. Now here we have something called the genetic code, and that's what we see over here on the right. There is a variation of this in Appendix B, I believe, of your textbook. Let me check. I'm sorry, Appendix D of your textbook has a variation of this. All this is, and I know it looks a little intimidating, you do not have to memorize it, you do not have to know it, you just have to know how to use it, and that's what we're going to do in a minute. This chart shows us all of the possible codons of messenger RNA. That's what these 3 letters on the left in each box are. We see UUU, or AAU, or CGC. So those are codons. And then they're attached to some other 3-letter combination, ARG for arginine, or SER for serine, PHE for phenylalanine. These are the amino acids. So let's go over here. We see in our just example that we draw, we have UGG is our mRNA codon that we have with a box drawn around it, UGG. We come over here to this box and we look up UGG, and that's what I see right there. What does UGG say? It codes for tryptophan, TRP. You do not need to know what the abbreviations for the amino acids mean. Sometimes I have to kind of reference because I forget, because you will never need to know that. All right, so that's mRNA. The next type of RNA we will discuss in a moment is transfer RNA, tRNA. tRNA are short lumps of RNA whose job it is to go and grab the amino acids that the mRNA codes for. And it does that using something called anticodons. Anticodons are the opposite 3 letters from codons. Let's see what that means. tRNA sometimes is shaped almost like a weird little guitar or upside down 3-leaf clover or something like that. And this is actually just folded up RNA. And down at the end where the little lumps are, there's going to be some exposed letters of RNA, some exposed nucleotides. Well, up at the top, let's go back. We had ACC was our triplet of DNA. And then our codon for messenger RNA was UGG. Well, the anticodon would be the opposite of these three letters. A, C, C. So many times it will be the same thing that was on the DNA. But an anticodon is going to be on the transfer RNA. It's going to be three letters of transfer RNA that correspond with the codon in messenger RNA. We'll see why that's important in just a moment. And the final type that we will discuss is rRNA, or ribosomal RNA. rRNA paired with certain proteins, well, that's what ribosomes are made from. Ribosomes are made from rRNA and certain proteins. And what did ribosomes do? Ribosomes built protein. Now, let's go ahead and start putting all of this together and see how protein is made. Well, protein synthesis, which is what we call the act of making protein, protein synthesis has two parts to it. The first is called transcription. Transcription takes place inside the nucleus. Transcription takes place inside the nucleus. And transcription is the act of reading DNA and making messenger RNA from it. Reading DNA and making messenger RNA from it. Translation, on the other hand, that's the second half. Translation takes place out in the cytoplasm at a ribosome. Translation takes place in the cytoplasm at a ribosome. Now, this ribosome could be floating around free in the cytoplasm, or it could be bound to a rough endoplasmic reticulum. But the process is going to be the same. Translation, a ribosome reads the messenger RNA and builds the protein from it. So let's see how that happens. Let's say this DNA contains a gene. This is just a segment of DNA that I tell you contains a gene. And the letters here, they're just letters that I made up. There's no significance to these letters. This is just the example that we're using. So this DNA contains a known gene, and we're going to make some protein. We're going to make the gene, or we're going to make the protein that the gene says to make. So the first step, transcription, which is what we're going to do now, is going to take place where? In the nucleus. The DNA is going to unzip from its other half. Remember, DNA is that double-stranded, double helix shape? Well, we're going to pull away one of those strands to expose the area where the gene is found. And then we're going to build our messenger RNA off of it. How do we do that? Well, we just take the base pairs. We're going to take the C in DNA and lay down a G. Take the G in DNA and lay down a C. And we're going to do that all the way down the entire length here. Now, you should be able to do this. If you were given this stretch of DNA, you should be able to write in the messenger RNA. And that's just from following the base pair rules. So that's transcription. Well, let's look to see why transcription is important. Well, what is DNA? DNA, we just said, contains genes, And genes tell our bodies which protein to make and when to make it. Where is DNA found? DNA is found inside the nucleus. Who makes protein? Ribosomes. Where are ribosomes found? They're found out in the cytoplasm, either free or bound to the rough ER. So DNA, in order to be protected, it stays inside the nucleus. It cannot leave the nucleus. The nucleus actually physically is not permeable to DNA. So DNA cannot leave. But ribosomes, in their functional state, they cannot get into the nucleus. So we have the recipe for protein in the nucleus and the guys who make the protein outside of the nucleus. We need to be able to communicate between the two. So that's why we make messenger RNA. Messenger RNA can leave the nucleus. So after transcription, after we've made our messenger RNA, the messenger RNA strand falls off and leaves the nucleus through one of those nuclear pores. And then the DNA zips itself back up, back to its double-stranded form. Now the messenger RNA leaves the nucleus, it goes out to find a ribosome, and the ribosome is going to attach at one end of the mRNA, and it knows which end to attach to. We won't worry about why. It knows which end to attach to. And the ribosome is going to read 3 letters at a time. It's going to read GCG, and then it's going to read CGU, and it's going to keep reading codons. It's going to read this first codon, and then the next codon, and the next codon. But the weird thing is, when it reads GCG, nothing happens. And it moves on to the next codon. And it reads CGU. And again, nothing happens. And it moves on to the next codon. And it could read many letters before anything at all happens. Why is that? Well, let's go back to our genetic code chart. If we look at these, almost all of these have an amino acid next to it. But if we look down here, this green box, AUG, it says 2 things next to it. It says MET, which is methionine, an amino acid, or START. Well, AUG actually is the universal on switch for a ribosome. All living things that we have discovered need AUG to turn the ribosome on. So when we read GCG, the ribosome isn't on yet, it just slides along. We read CGU, the ribosome isn't on yet, it just slides along. And then we get to AUG, and that tells the ribosome, start making protein here. And what else does the ribosome do? It lays down methionine. Methionine is always going to be the first amino acid laid down. It may later be removed if we don't need it, but AUG says start making protein here and lay down methionine. And now we're going to read 3 letters at a time from this point, and we're going to look those 3 letters up on this chart and lay down whichever amino acid it says. So we're going to read AUG, lay down methionine. We're going to read ACU and lay down threonine. We're going to read GGU and lay down glycine. CGA, lay down arginine. AUG, well, didn't we say that means start and methionine? Well, when it's the first one, yes. But we could have AUG any number of times throughout. Once we've already started, all it says is methionine. We don't start again. We're already going. So AUG here says methionine. And we would keep going, a couple of hundred to a couple of thousand amino acids long. This is just a really abbreviated one. And we would keep going until we get to, let's go back to our chart. We have these three red boxes, UAA, UAG, or UGA. All three of these tell the ribosome, stop making protein here. And the ribosome actually falls apart so that it can't make any more protein. So when we get to UAA, stop. Now there's going to be more messenger RNA, but we ignore it. The ribosome doesn't exist there anymore. So now we have amino acid sequence. A whole strand of amino acids, like I said, a couple of hundred to a couple of thousand. What do we call that? What do we call this sequence of amino acids? Think back to chapter 2. Remember, that is the primary structure of a protein. We don't have a protein yet, but we're on our way to making it. So let's pause this and go back to see, why is there this big gap here? We've left something out. And then after we talk about that, we will go back down and look at our primary structure. That big gap is where our transfer RNA is. Let's look at why this is important and why we jumped over it in the first place. Well, we have our anticodons, the three letters of transfer RNA. These are the guys that go and grab the amino acids and bring it back to the ribosome to lay it down. And we know that they are the anticodons because they base pair with our messenger RNA. So if the transfer RNA matches with the mRNA and then lays down the amino acid, why did we skip over that step? Well, I do that just to kind of make sure that I do the right thing. Let's see why I did that. UAC or UGA or CCA, all of these use the same four-letter alphabet, A, C, G, and U, that messenger RNA uses. It would be really easy to look up UAC on my chart here. But if I look up UAC, that's tyrosine. That is not methionine. So to make sure that I don't accidentally look up the tRNA on my chart, I add that step in last. That way I only look up the messenger RNA, lay down my amino acids, and get my primary structure. Okay, you can do it in any order that you want. I just do it that way because it keeps me from making mistakes. All right, well now we have our amino acid sequence, our primary structure. Here's what that whole thing looks like in action. Here's my DNA inside the nucleus. Here's that area where the DNA unzipped. Here I'm making my messenger RNA off of it. The messenger RNA breaks off and it leaves through a nuclear pore. Now my messenger RNA goes and finds a ribosome, and here this little guitar-shaped thing has my three anticodons that match up with the three codons on the amino acid. The ribosome slides along. AUG, well, there's my methionine. This is also the on switch. My next three letters, AUC, well, here comes the next transfer RNA carrying an amino acid. There's the anticodons, UAC. Oh no, I'm sorry, that's the one that we see right here. The next one would be UAG. And then we would keep sliding along 3 letters at a time, adding more and more and more amino acids. That chain of amino acids keeps growing until eventually the chain is finished. This is my primary structure. The ribosome, meanwhile, falls apart. It's done its job, it's finished. And up here at the top, this should look familiar. This is something that we looked at back in chapter 2. Here's my primary structure. It's just amino acid, amino acid, amino acid. How do I know which amino acids? Well, the DNA told the mRNA told the ribosome. So it's a very specific primary structure. And then based on that primary structure, it folds up or coils up into the secondary structure, which folds up even more into the tertiary structure, the three-dimensional shape. And now we have a protein. Okay? Sometimes multiple proteins join together to make a quaternary structure, but not all of them did that. Well, this is kind of a messy business. There's a lot going on inside of a cell, and as that chain of amino acids grows, sometimes it starts folding up, and it does so a little prematurely. Maybe it folds up incorrectly. Well, luckily, we have something called chaperone proteins. Well, chaperone proteins guide the folding. So as that strand of amino acids is emerging from the ribosome, the chaperone proteins help to guide it as it folds up to make sure that it folds into the correct shape. Remember, with proteins, shape is everything. Maybe it still doesn't fold correctly. And if a protein has not got the right shape, it can't do its job, remember? So a proteasome is a special protein that breaks down misfolded proteins so that they can go through the process again and be assisted by a chaperone protein. But proteasomes just break down misfolded proteins so that they can try to go through the process again. Now, maybe that happens loose in the cytoplasm. Maybe it's going to go through, here's our rough endoplasmic reticulum. We saw a picture very similar to this at the end of chapter 3. Going from the rough endoplasmic reticulum through a vesicle to the Golgi, through another vesicle either as a lysosome, a peroxisome. Maybe it's going to be secreted, maybe it's going to become a part of the cell membrane. But where did that protein come from? A ribosome. How did the ribosome know what to make? The messenger RNA told it to. How did the messenger RNA come about? Because of DNA. All right. Now let's change gears and talk about cell division and the life cycle of a cell, the cell cycle. And that's what we see here. We are not going to go into a whole lot of detail, and I've actually included some videos to kind of guide through this, and you've already talked about it in lab. So what we see here, this is called the cell cycle, and this just kind of shows us the life of a cell and the different stages that it goes through. And we're going to start right here at the end of this brown arrow. A cell has just been created. How is the cell created? Through cell division. Remember back at the start of chapter 3 when we talked about the cell theory? All cells come from pre-existing cells. Well, here this cell came from a pre-existing cell and now it's beginning its life. It is in something called G1 or growth 1. Technically it stands for gap 1. But I like to call it growth because that tells us what's it doing. When it's first made, it's kind of small. It needs to get a little bit bigger. So during G1, that cell is going to grow a little bit. And most of the time, we don't need to make more cells. As a matter of fact, the only time we need to make more cells is if there was damage and we need to replace those cells, or if we are not fully grown and we need to make more cells to get bigger. But once you have finished growing, most of the cells in your body are not going to divide that often at all. So once we get back to our full size, we actually leave the cell cycle entirely into something called G0, or G with a little 0 next to it. During G0, that's just a cell living its life, doing its things that it's supposed to do to keep you alive. But if a cell needs to divide, either because you are still actively growing or because we need to replace some dead cells, well, then we come back into the cell cycle. And once a cell is ready to go, once it is at the size that it needs to be, we move to the next phase called the S phase. The S phase stands for synthesis of DNA. This is where we take that DNA that we talked about, those 23 chromosomes, and make exact copies of them. We're going to synthesize a full other set of DNA. Once we have done that, we need to grow a little bit more. We're go into something called G2 or growth 2. Really, it's gap 2. We're going to grow a little bit more. We're going to duplicate all of our other organelles, because if we're going to make 2 cells, we have to have 2 of everything. Down here in the S phase, we duplicated our DNA. In G2, we're going to duplicate everything else. Now, all of that, from the time this cell was born until right before we start to go into cell division again, that is all something called interphase. Interphase contains G1, S, and G2. We can kind of say that it contains G0 also, even though G0 or G0 is not a part of the cell cycle at all. But interphase is when a cell is not actively dividing. And most of the cells in your body are in interphase. Really, they're in G0. Now, once we've duplicated everything, it's time to go through cell division, mitosis and cytokinesis. And mitosis is the division of the DNA. Cytokinesis is the division of the cytoplasm. They occur at the same time. Mitosis starts first, cytokinesis starts a little bit later, but they overlap. Cytokinesis typically starts partway through anaphase. But mitosis is the division of the DNA. And it has different steps: prophase, metaphase, anaphase, and telophase. The cytokinesis portion is just division of all of the other non-DNA components of the cell. Once we have completed telophase and cytokinesis, we're right back where we started, but now we have 2 brand new cells. If those 2 brand new cells are all that's needed, they leave and go into G0. If we still need to make a few more cells, we go through this whole thing again. And we keep going through this whole thing again as long as we need to produce more cells. Now, this could be a tricky thing, because we don't want too many cells, and we need to make sure that the cells that are going to go through mitosis are healthy. So, how do we make sure? Well, at the end of each phase, there's a checkpoint. It's essentially the cell looking at itself and saying, okay, I've done everything I need to do in this phase. Let's make sure we're okay. And now we go to the next phase. So at the end of G1, the cell checks itself before it goes into S. At the end of S, the cell checks itself before it goes into G2. And right before mitosis, the cell checks itself to make sure it's okay before we make a duplicate. Why does the cell need to check itself? Well, 1, to make sure that it has everything that it needs. But 2, let's look right at the end of G2. What if this cell has something very wrong with it? Maybe cancer. What happens if we make a duplicate of that cell? Well, now instead of 1 cell with cancer, what do we have? 2 cells with cancer. Because mitosis makes 2 exact copies of the cell that we started with. Everything about that cell has just been duplicated. So if a cell has cancer and it goes through mitosis, now we have 2 cells with cancer. So the cell has checkpoints to make sure not only do we have everything we need, but is this cell healthy? Well, there's something called the p53 gene, and you probably don't find this in your textbook, but the p53 gene is sometimes called the guardian of the cell cycle clock. And it is this cell that either allows or prevents movement from one phase to the next and ultimately into mitosis. If something is wrong, the p53 gene will not allow for mitosis to occur. But obviously cancers do happen. So what's happened? Well, if there is something that interferes with p53, what we would call a successful cancer, which doesn't sound like something we would really use, but a successful cancer just means a cancer that is proceeding as cancer. In a successful cancer, something has managed to bypass the p53 gene. The p53 gene can be deactivated so that a cancerous cell is free to keep going and going and going. Well, let's define cancer while we talked about it, because we've said it a few times in this course now, and we're going to talk about it quite a bit this semester. Really, all cancer is mitosis when we don't want mitosis. Because, like I said, we should only have mitosis if we are still actively growing or if we are repairing damage. No other times should there be any mitosis. But cancer is just mitosis when we don't need it. So here's another drawing looking just specifically at mitosis and showing what happens at each phase. And here's a video. I'm not going to play this video. It is included in your PowerPoint on Blackboard, so you can watch this video where it shows what happens during mitosis. And again. So now let's move on and talk about something else that cells can do, and that's called apoptosis, or sometimes you'll hear it called apoptosis. But apoptosis, which is how it's more normally pronounced, is something called programmed cell death. Programmed cell death. Oftentimes you will often hear it just called cellular suicide. And cells are constantly checking themselves, not just during the cell cycle when it's moving from one phase to the next, but cells actually regularly check themselves just to make sure everything is working as it should. When something is found that is unhealthy for the cell, or if the cell has just taken too much damage, when the cell has lived its life and needs to be replaced, there are mechanisms inside the cell that cause the cell to essentially kill itself. And it can do this a few different ways. One common way is it builds up a bunch of lysosomes and then they rupture, spilling all of those digestive enzymes into the interior of the cell, and that breaks the cell down. But there's a few mechanisms for apoptosis. But apoptosis is something that the cell does intentionally. And there's a gene called PRB. And PRB actually is the gene that triggers apoptosis. And really the way that it works is a cell is constantly building up ready to cause apoptosis, and then a check comes along and your cell has a feedback that says, okay, everything's going fine, do not proceed with apoptosis. And then we start to build up again towards apoptosis, and another check occurs, and feedback happens that says, no, everything's okay, do not proceed with apoptosis. If something goes wrong inside the cell, then that feedback doesn't happen. We're building up towards apoptosis, the cell recognizes something is wrong, we do not get that feedback, So apoptosis proceeds and the cell kills itself, which is a good thing because let's say something occurs inside the cell and it is on the verge of becoming cancerous. Well, if the cell does not fix itself, if it does not kill itself, then that cell will go through division, mitosis. And now we have 2 cancer cells, quickly 4, quickly 8, and they just keep dividing. And apoptosis could have prevented that. But why didn't apoptosis prevent cancers? Well, again, if something can bypass the PRB gene, if it can deactivate the PRB gene, well, then we got a way around the whole apoptosis thing, and the cancer can proceed. So apoptosis is programmed cell death, and it's not a bad thing. PRB is the gene that controls apoptosis by initiating apoptosis. But in some cancers, PRB has been kind of turned off and apoptosis has been prevented. So cancer, as we said, is just runaway mitosis. It's mitosis when there should not be mitosis. How do we know when a cell needs to divide versus when it doesn't? How do we know if there's damage or if we're growing? Well, cells have a couple of things. One, we say they are anchorage dependent, and 2, they are contact dependent. Cells need to be anchored to their surroundings, and cells need to be in contact with a certain number of other cells depending on which type of cell they are. When they are anchored correctly and when they are in contact with the proper number of cells, there is no need for mitosis. Let's say you get a cut on your arm. Well, we have just broken some of the anchorage, and the cells are no longer in contact with the correct number of cells. So they start going through mitosis, 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 until they are back anchored to the correct spaces and in contact with the correct number of cells. Once that's in place, P53 says, okay, now we stop. Cancer means that not only have we bypassed P53 and PRB, we have also bypassed anchorage and contact dependence, because in cancer, we have too much contact, but we still keep dividing. And we're not really paying attention to whether or not those cells are anchored. Let's use a thinking cap question. Why will there never be a cure for cancer? And similarly, why can we safely say that anything purporting to be a cure for cancer is a scam product? If you ever see something that says such and such is a cure for cancer, or the doctors don't want you to know about this cure for cancer, how do we know that those are scams? Well, the thing about cancer is it is not a disease. Cancer is a group of hundreds of diseases. Now, there may very well be a cure for small cell lung carcinoma, or there may very well be a cure for a certain type of glioblastoma, or something like that. Now, eventually, hopefully, we will have a cure for every type of cancer, but there will be lots of cures. It will not be a single cure for cancer as a whole. And that's just because the only real thing that cancer has in common is that it is mitosis. And we cannot turn off mitosis, because mitosis is needed to replace damage that occurs in our body all the time. So we can't just stop mitosis. If you ever see something that says it is a cure for cancer, you can be absolutely sure that it is a scam. Now, maybe it might very well cure melanoma, but something that cures melanoma is not going to cure a completely different type of cancer, because they do not have anything in common other than they are both runaway mitosis. Okay, so that is the only lecture for chapter 4. There's only one lecture for this chapter. The next time we will come back and talk about metabolism. And when we talk about metabolism, it's going to be specifically talking about the mitochondria. So refresh on that from chapter 3, and I will talk to you next time.